happen today. But that can be an ongoing discussion. But it would be useful, I think, for the folks that are more interested in water quality, for them to know if they should be working on something else, either adjacent to or in addition to the, let's say, a USDA grant for spring.
Right. Well, it might also be relevant even for the 30th when the idea request for money comes in, because
yes,
$1,000 should go to a separate group versus.
That's correct. Yeah, good point. I'll move that to the top of the See Superb item agenda then. Wrong part. Never mind. Water quality. There we go. Okay. So JGI grant. Hi, everybody that just joined us in the last couple of minutes. We're taking a look. I'm going to repaste because I don't think those of you that just joined, Greg, Franz, can you see what I pasted into the chat before you joined? I'm never really sure how that works. Does your chat window look blank, or is there something there? The point being that I created an agenda for today, and I've also got a separate document going now for, there, I'll just paste it into the chat again. Got an agenda for today and a separate document where I've moved the title and abstract away from where we were previously editing it to make a clean version to start fresh for review today. So if you take a look in the chat, you'll see the file names for those documents we can refer to during our conversation.
Joe, do you want us to open, are they in the Google Drive thing? Is that what we're looking at?
Now in the root folder on the Google Drive, yeah.
Okay.
Those two document names are there. Hello, whoever just joined us.
Okay.
So Joe, we almost have to have this conversation about water or water right away because it's in the project title. And obviously if we're not doing water, it'd have to go out of the project title.
Right. Let's do it. So,
well,
yeah.
I think like Jeroen mentioned it, and I commented on it on Slack. Since in the beginning on our second day in Fresno, we were kind of heavily on the algae component of the project. But then on the third day, kind of like diminished, and it just became like some kind of assessment for water quality, which I don't think is always a correlation between the two. And so it kind of got a little bit neglected. I still find it very interesting, the water component, and we could like make it into something bigger. I'm kind of like in between as a microbiologist. I don't know if you read this morning, I commented on the Slack again, that the soil thing is of course closer to where I stand as a microbiologist and plant pathologist. But I think since the group is big, I sometimes even wonder what my real contribution could be to the project, although I would like to be very much involved. So maybe if we could do 2 projects, but like Jeroen mentioned that the project could be closely related and there could be a lot of like things going back and forth between the 2. It would be nice if we have like one big group and each working on 2 sub-projects that maybe at some point will come back together. But that's not too much. That was our idea.
Yeah, I mean, I think that makes sense. The water part makes sense connected. But I think the question is what the actual, I guess, assay or measurements will be specific to that part. I think that's where we're struggling. Like I know the algal assays was the main focus. But I think there's like logistical and feasibility questions. I think unless we can figure those things out, it would be hard to.
Yeah, like the first thing that came into my mind, and I'm not a water expert or an algae expert, I'm a bacterial person. Like the guy, I forgot his name, who came in on the third day, said the water was highly salty, saline. And so there's diatoms, algae that can grow in that water, and even like tiny little steps are being made to using those to lower the water salinity. And so I was thinking more like, I mentioned it in Fresno at one point, like what you throw water onto the field, water is an issue. What if we can get like 2 things out of the same gallon of water, not just like for growing the plants, improving soil health, all that, what we have been discussing before, but then when the water runs off, we can collect it in some way and still use it for something else instead of just like being waste. And then we can use it to grow these specialized algae that can then be used for animal feed, biofuel production, or I don't know what else. And then the water might still be used to grow different type of algae, or then it becomes waste, but at least you have done something else with it instead of it just being waste. So there I see that it's like definitely 2 different projects, but they might be connected at some point, because if you can improve soil microbiome, that will benefit and crops and make the water less dirty when it comes out of the field, then the other project can like.
Right. So I think so I think the question is a logistical question. So maybe the project can be not to determine how we can actually collect the water properly for this purpose, right? So maybe we're not taking the full step into actually analyzing it as part of the project, but the question is, can we, like, what is the best way to collect it? And that could be a question.
Yeah.
And then the next step will be to interface it with, can we use that to then do these other, answer these other questions that you're bringing up? I think that part is, because that part's missing, it's hard to connect it, I think. Right?
Yeah, I understand the technical thing, but
can
you
the
can
technical
still
part
do it
be
like
the question?
from a scientific standpoint, and just like take these big water basins that are runoff, agricultural runoff from a couple of fields together. Okay, then you might not immediately connect it to like the soil microbiome, but that might be in a greenhouse, and then you can, like, you know, with engineers try to get it out into like a field component. Well, me, I'm not an engineer, I'm not a field scientist, I'm like a lab scientist going into a little bit of greenhouse studies. So yeah, I'm definitely not the one to have all the answers to that.
I mean, it seems to me there's definitely a project there, but it's already what you described is many faceted and could be a project in and of itself.
Yeah, that's what I mean. It's like, it's not nowhere, it doesn't have to come into this soil project, right? I'm just trying to see, we're just trying to see, are there other people that might be interested in it? Because the CSAP, okay, the $1,000, we have to be in one group and we have to go in the, but I think you should not be limited to just one NSF or USDA proposal. You could be in multiple, just not for CSAP, but it could still be like a nice outcome from the Ideas Lab. Now that we've all got to know each other, and I kind of really like this multi-CSU kind of thing, I think we should benefit from, you know, we're still connecting with one another to exploit our interactions, because once we're going away from workshops and all, and we say we're going to keep contact and we're going to collaborate, it usually doesn't happen. I just want to like use that momentum, you know, to do something now that we finally find some people that have some similar interests, although we still haven't quite figured out how. I would really like to keep going, because so many times I say, yes, we will do this, and then it never happens, and I find that kind of, you know, and if it doesn't happen, that's fine too, you know. It's just like, just wanted to throw it out there.
So Isolde, I agree with you, but on the other hand, from a pragmatic perspective, I think it's more important to get the urgent steps that we need to take.
Of course, and we need to go. We need to go ahead with the soil project, definitely, and this should be our top thing. We just wanted to throw it out there that, you know, if somebody wanted to, we're very, you know, eager to get this other project started too, but for now the focus has to be on the other one. I clearly agree. Yes.
And I believe once we have the okay from Susan to go ahead with the larger project, we can easily internally decide, hey, let's pursue this other one either instead or on the side. That's, I would say that's up to us.
Excellent.
Agreed.
I think if I can weigh in on a little bit, having to try to search for the Google Drive and everything while this was going on. But I think there might be two questions in this that we could deal with at the February meeting. So it might be that if people hang on and we kind of work out this soil component, and the people that are interested in water work on that and see if there are any ways that there might be linkages or synergies that could be sort of leveraged, then maybe at the February meeting, the projects might split and there might be two proposals that come out of it. And that might not be a bad thing, but it would also allow everyone who's interested in this general area that we've been talking about for the last 2 weeks to kind of still both benefit the project, whichever project you're talking about, benefit that, but also benefit from just the exchanges that are going on and the knowledge that's being exchanged at this moment. So I would sort of,
I think that is an excellent idea.
Propose that everybody apply to go to that meeting, because otherwise you're not going, right? And then you're out. And that's fine if you want to do that. Everyone has to make their decisions, but you know, I'm kind of hanging on to see how, and trying to contribute in ways that are constructive. And because I'm learning like I haven't learned in about the whole time I've been an associate professor from this group. And so for me, I don't know where this is going, but I'm trying to just contribute in a way that I think I can do it. And like that email that Susan sent out earlier, you know, this project is meant to stretch everybody in ways that they haven't been stretched before. And NSF is looking for stuff that you're not staying in your wheelhouse and like going and writing about something that you already know about, that you're really trying to contribute to something that's new. And so the water quality stuff, from what I've understood, is very, very interesting. And Tom, I think, is the name of that dean. I think that's his name, the water dude at Fresno State. There's a lot of stuff going on about water in the Central Valley, and his stuff is super interesting, and his talk was inspiring. That's not going away. And if there's a way that this group can kind of spin off into like a project that would then, you know, really illuminate that dimension of the ag sector in the Central Valley, or look at that, the science of it, right? Not just the politics. I mean, there's tons of stuff about the politics and who really, you know, what a nightmare, right? So, but I think that if everybody hangs in there, there might be two projects, and it might not be a bad thing. And, you know,
yeah, I think that is perfect. We don't have to decide this now. There's some time between now and January, so we can read and inform ourselves a little bit better, so we can have like some more, you know, like answers to like the most urgent questions, and then we can discuss that, and then we could still decide.
Yeah.
So I'm seeing lots of nodding. So it seems to me then that the group stays together at this point. We all submit as one big group. In January, when we meet, the folks that are potentially interested in water and looking at that could come with some ideas, and at that point we could decide whether or not to split it off into groups.
Yeah, I think that's a good summary.
I have another proposal contradicting my own suggestion before to focus on the imminent stuff. So there are a few people here at Cal Poly looking at Agricultural Research Institute grants. And one of the ideas that we have is to look at water aspects, specifically for the Cal Poly Properties Swanton Pacific Ranch. I don't know if you know about that. Cal Poly has a ranch near Santa Cruz, and that has to be maintained for research purposes. And one problem they have is salinity in the water. And so we're considering proposing an ARI project on water in general. So once we decide if we're going to go ahead with this, I'm certainly interested in getting people from this group involved as well.
Excellent.
Sounds good.
So it sounds like we keep water in the title of the project abstract for Sea Superb.
Yep.
And then we'll
I
see.
would keep it in there, yeah.
Yeah, it sounds like there's two things. We got to keep two things in mind here. There's the JGI thing that has to happen like tomorrow, right? That's like the main thing. But the other big important thing that we can't forget about, and that we kind of have to kind of figure out, and it's going to evolve, right, is the Sea Superb thing due on Monday or whatever, on the 30th. And that's harder. Like this JGI one is really focused on a really narrow dimension of the project. And it's super important for us to have that going. But the bigger thing and the more interdisciplinary thing, and the thing we really have to kind of expand our heads around is this other integrative project. And that's not going to become clear at least until January. And so I think people have to be uncomfortable with that. And I'm sorry, they have to be comfortable with their uncomfortableness about this so that we can keep it moving forward. And things are going to, they're going to change. It's going to be interesting. And I think that's what people have to keep it like in the back of their head. We don't know where it's going. That's a cool thing, So
I think the project title and the abstract actually look great for Sea Superb, keeping in mind that it doesn't have details about water and it doesn't have details about potential farmer grower interviews. I don't think it has to be comprehensive. I think it just needs to give Susan, the Sea Superb program office, some idea of what we're doing. And I think what we have accomplishes that quite nicely. So rather than probably spending a lot of time trying to craft that to a beautiful and comprehensive document, I would just say let's go with what we have, Joe. It looks beautiful. Thank you for all the work you put into that. And maybe we focus on the JGI proposal, because that actually does have to be a finished document, and that's not just going internally to Sea Superb. That's going to a funding agency.
Sounds good to me.
So if we follow that line of argument, then we move on to the next part of the agenda, which is the JGI grant.
Okay.
I've heard from email, I think the whole group maybe, or at least some subgroup of us have heard by email from a few people saying that they are or are not interested in being co-PIs on that grant. And so that first bullet point is there's a CV folder on Google Drive. If you want to be a co-PI, drop your short form CV into that folder on Google Drive, and that's how I'll know who's opting in to be a co-PI.
Could I ask a question about that?
Go for it.
Do you think I need to be a co-PI on that?
I don't even need to be a co-PI on it.
I mean, who should be on it?
Administrative. Yeah, I don't know. Like I wrote yesterday, I think, in email to everybody, there's no real valor in being a PI on it because there's no money changing hands. You could put it on a CV, but it's not going to do anything RTP wise for anybody, or I think it's just more administrative work.
So the question is, maybe what gives the strongest application, I guess, is really the expertise, right? So it's all the right expertise in this grant is what I would suggest. So whoever has expertise in this area, which I think is, definitely Nate and Bill. Other than that, I don't know who does, maybe Matt as well, doing the metabolomics and metagenomics stuff.
No, I think it's got to be, at least for the PI should be either Nate or Bill, and then the rest of us as co-PIs is fine. I think it matters really. The rest of the co-PIs don't have to have specific expertise, but the PI should be one of those two.
Okay.
I noticed in the review criteria, it was, some component of it was, do they think that the PI can use the data and analyze it and take significant conclusions from it. And I'm guessing that Nate and Bill's are going to look more like that will be the case than my publication record.
Okay.
Yeah.
So Joe, just to clarify here, in order to serve as a co-PI on the JGI grant, is it enough to collect the soil samples, extract the DNA, and send it to you guys for sequencing? Does that qualify me to serve as a co-PI?
Sure.
Okay.
Or collect samples. I didn't see any advice on what co-PI status indicates, but I think that John maybe was right that it should be a collection of people that have a vested in or a demonstrated interest and expertise in the process.
Yeah.
So I might not even be a co-PI. I'm just sort of administratively organizing as best I can, but
No, you'd have to be co-PI because you're going to be doing the DNA isolations.
Oh, fair enough.
Yeah.
Yeah.
I mean, do I qualify if you use my dirt?
Yes. I mean, they're not gonna nitpick the co-PIs and ask what is each co-PI doing, because I mean, really, it doesn't matter. They're not giving us any money.
Right.
There can be 100 co-PIs or one, and it's the same for them.
I nominate myself to be the last co-PI.
Well, we're going to have to pass. I agree with you that either Nate or Bill should probably be the PI, which means they're going to be the ones that need to submit all this information. And it's too bad that neither of them are able to be here right now. So I will do my best to pass all of this information on to them.
Joe, have you actually tried to open up the submission form? Is it just boxes that you paste all this information in?
It is. It's one long single-page web form.
Okay.
There's a Bill sent out a version of it in an email to us all a week ago or so, and I've dropped a PDF printout of the web page on Google Drive at some point. It's in the JGI folder.
Okay.
Not a very good printout, though. It's missing some of the content, but it gives you an idea of what's there. And there are some details that somebody needs to figure out, and that's part of the rest of the agenda for this grant for today, if we have time to look through it. With it. Oh, neat. Excellent. Good timing.
Yeah, I didn't put myself on as co-PI because I have my heavy teaching schedule now, and I wasn't able to contribute, so I don't feel I should be on there. But I'm willing to provide samples or whatever you guys need, so no worries about that.
From my side, to be honest, I don't see many contributions that I could make. Maybe once you have the data, we can do some AI analysis, but then Bill is much better equipped to do that. I'm certainly not in a position to easily collect samples. I wouldn't know who to approach in the ag department.
Mm-hmm.
Well, it's probably just fair to say that if your CV is in the Drive folder or you email Joe with your CV, then you can be a co-PI, and if you don't, you won't.
That's the idea.
Okay.
Yep. That's how we're going to hopefully, and if you have other questions that you want to talk through whether or not to be a co-PI, that's fine. But yeah, ultimately that's going to be the black and white. If you decide to opt in, make sure your CV is on the Drive folder or you've emailed it to me by tonight.
So I had a question for that too, Joe.
Hey, you're the PI now, by the way.
Yeah, we decided.
So one trip to the tomato field, and I'm the PI, huh?
Right.
For this thing, is it supposed to be a full CV or just an abbreviated one?
It says short form with no other details.
Okay,
so
I got probably like a 5 and 5. Okay.
Somebody sent me their NSF short form version. I might turn in my NIH 6-page bios. I don't know, something like that.
Okay.
They didn't give, at least in the specific call, they didn't give direction as to what they meant by short form. The reason, so I'm glad you were able to join us now, Nate. We're all glad. We were discussing who should be PIs, co-PIs on the JGI submission tomorrow. And there's been some discussion about whether or not the PI should be the person with the publication record that's most applicable to what we're proposing to do. So I had been, since I've been collecting all the data, I was planning on just filling in the form and submitting it myself, but that would make me the PI, and I do not have a track record of research that has anything to do with metabolomics or metagenomics.
Yeah,
so
I mean,
we discussed
sorry.
would be the best choice for filling in the web form and pushing the submit button.
Yeah, sorry.
Go ahead.
I'm happy to do it, but I'll say that, like, my CV right now is like, we're in the middle of this grant, so we haven't published a lot of stuff. So we have the grant to support it. This says that we do the work, but I don't have a lot of the publications yet out for that specific stuff, right? So I'll just say that right up front. Like, I've got lots of genomic work on there, but in terms of microbiome stuff, we're in the middle of doing those projects. So we haven't published all that work yet, but we do have the grant to support the work if that's helpful. The other person would be Bill, right? So Bill's got,
Yep.
He used to work at JGI, so that's another way to go. But I'm happy to do whatever the group feels is best.
Any other discussion on that? I'm not sure how they'll read it. It's a request for a small number of samples being processed. I'm not sure how carefully they're going to look at CVs.
We've applied for them twice before in the past with varying success. Like, we haven't gotten it yet, but like, it wasn't clear to me why. Like, it wasn't that like, I just think it had more to do with the group you were competing against and when you were competing against them with.
Yeah.
I mean, it's a new investigator program, so it's not meant to be established people.
Right.
In some ways, right? So.
Yeah. Do you think, I guess the only other thing that I would contribute to this discussion at this point about who should be the PI is, Nate, do you intend to be a PI? If you are the PI on this and you get it, then that makes you ineligible for real JGI grants as a new investigator status.
Oh, it's fine. That's okay. Because, part of it is too, like, there's benefits to being in the CSP also. So, it's fine. I'm not worried about losing my new investigator status.
Okay.
So I'd be happy to offer that to the group.
Thanks for your sacrifice.
Wow.
Yeah. Yeah. It's a big one, I know. It's a big one.
Okay. So it sounds like, do we have any other discussion about that?
So just so I'm clear, the deadline is tomorrow, correct?
Tomorrow at 11:59 p.m. Yeah.
Okay. So I'll have to go through. I put in my component last night. I haven't kind of has anyone looked to reconcile like the entire document yet?
I've looked over it a bit this morning, but I'm suggesting rather, that everybody look over it by the end of the day today. Right? Final comments, suggestions, and that gives all day tomorrow and Friday. Sorry, all day tomorrow to copy and paste from a Google Doc into the web form.
And relevant references.
Right, so I did stick in a few references. There's no separate space for references. You just have to put them in as part of the character limits for each of the text boxes.
Oh, okay.
I'm trying to remember how I did this last time, because I feel like.
And you will have to create an account to be able to see the submission form and populate it.
Yeah, I already have an account there. I've submitted to this program before, so
Yep.
Unsuccessfully.
Yeah.
So you're putting your trust in me,
yep.
Who's already been unsuccessful with them. Although I guess we've all probably been unsuccessful with various grant proposals in the past. So
since you've been in the tomato field, you might want to look over. In fact, I'm going to paste this into the chat again. Let's see, there's one other document. I made a new document just with the, oh no, I don't, never mind. I'm thinking about different documents. So the document we have been co-editing, I filled in a few highlights, some yellow highlighting on details of the sampling that I wasn't sure about since you just got back from the tomato field. So there are still a few things at least to discuss in the JGI draft.
Yeah, and I'll also say, so what I did last time is I submitted, it looks like that the attachments were abbreviated CVs that I had to submit, and I just submitted a PDF of references. So there was a way for me to attach a supporting document.
Oh, okay.
So, so what I would suggest is that we footnote it, and then just include a reference, set of references as footnotes.
Yeah, it looks like everybody has put their citations, although there are only a few of them, in sort of at the bottom of each text box. So those could easily be cut and pasted together into a single cohesive document. It looks like there are maybe 6 or 7 references throughout all of the current versions of the statements. So they're not that many.
Yeah, I think I had about like 26 last time I saw it. But, you know.
That might hurt us too, but.
It is what it is, right? So like, I mean, put in whatever you want. But that's, I don't really, I mean, I'm not all that worried about it.
Yeah.
So, but I think I will need, you'll have copies of those CVs, because I might need those CVs.
Yeah, so we're dropping all of our CVs onto the Google Drive. There's a folder for them in the JGI folder.
Okay.
So those should be up by everybody. Everyone who opts in to be a co-PI is going to put their CV in there by tonight.
And just so I'm clear, I had started to pull together text on the, like what the sampling distribution would look like. Where is it? Like I closed it. Great for
I hate it when that happens.
Where's that stupid document? Sorry, I gotta find it.
That's okay.
Just give me 1 second.
So for the rest of you, while Nate's working on that, if people can jump in at some point today and review those different statements for the JGI submission. Most of them are pretty short, mostly because I wrote them and like I said, I didn't have a ton of time to work on each of them. So if you do think of other aspects of those that should be flushed out a little bit, please do. There's plenty of, except for one of the statements, there's still plenty of character count left, which could work for us or could work against us, because as we know, the reviewers don't want to read a bunch of text that they don't need to. And the instructions do say briefly explain, and then give us a 4,000 character limit. So there's a balance there perhaps to be struck. If anybody particularly has time today, the ones that I'm most concerned about, having never done this before, and maybe Nate has more insight into this, is the sections on, let me see, the DOE mission, how the project relates specifically to the Department of Energy mission. I drafted this morning, we had talked about seeing if Bill would do that, and I emailed him about that, but I haven't heard from him. So when I was up at 4 this morning, I decided to jot something down. So that one I think maybe needs help if you have particular time to work on that. The other thing that I had overlooked, and again, Nate's probably had to fill this out before, so maybe Nate already has boilerplate on this, is there's a component, the samples regulatory compliance comment, something about transferring organisms from place to place. So if anybody has any input about that, that might be useful. Yeah.
I think there's only limitations between states, not within state, right?
Yeah, it just says utilization of any genetic resource may be covered by local, national, or international regulation, for example, as required by the Convention on Biological Diversity, Nagoya Protocol on Access and Benefit Sharing. This requirement applies whether the use is nonprofit or for-profit. Please provide a brief description of how you're addressing these requirements. So maybe we just type in not applicable, but yeah, we've got 4,000 characters. So yeah, we're not shipping them between states, between countries. But
I think that's probably all we need to say is that these are not genetically modified. We're not.
They're not commercially useful.
We're not culturing them.
Right.
We're not.
There's something about cultural appropriation in that big policy too. So something about we're not stealing these from native peoples who have a particular interest in them.
Yeah, we can say that they're either coming from public or private lands. And I don't think that there are any distinct things that we're doing. That's, yeah, we're not going to leaving the, so Nagoya is, making sure they don't leave the country. So it's not international. I think we're fine. We just say that. We'll be shipping them from within the state. They come to us with permission of use from owners of the lands they're from.
Yeah.
And then we should be good.
They're not genetically modified.
Yeah, they're not genetically modified or cultured. It's just a soil sample.
So where are the final samples being sent to? Like, where's GGI?
Berkeley.
Yeah.
Okay, so it's still local. Okay.
Yeah. They might send, I'm actually not sure where they may send the metabolomic stuff because I thought they were sending it to Arizona. But, you know, I'm not sure. I'm not sure if they're doing it in-house or not.
So really quick question regarding the metagenomics. How many samples are we talking about? Like each one of us who's a co-PI, how many soil samples are we supposed to collect and send them off to you?
Yeah, I think we were only talking about collecting at 2 sites, right? Nate, you were going to collect at a tomato field, and Greg and I were going to collect at a tomato field at San Marcos, correct?
Yeah. So let me give you some details about what I did yesterday, and then maybe that will help us understand scale and what you want to do. So I collected more than we needed yesterday, just because I was there, right? It was from a Roma tomato field. What I did is I collected samples that were, is there like a, I wonder if I can do this in the chat. I have a couple photos that I can share. Chat.
You can also share screen if you want to, but.
Oh yeah, I can just share screen.
I think you can.
Let's see if I can open this up. But basically, like I took a soil sample, like a trowel length away and a trowel length down, and like 4 inches down.
Nate, was it directly below the plant so you get root as well, or was it somewhat?
To the side of the plant? So,
okay.
So there should be minimal root there.
Okay, so not under the kind of canopy of it. Okay, got it. Mm-hmm.
Yeah.
I've got a question about this too. I've got a tomato hornworm infestation, and I'm wondering if do we want to make sure that all of their poop is not in the sample?
Yeah.
Right?
Yeah.
Okay.
Good luck with that.
I mean, they're like giant, prehistoric.
Here, let me share. I'll share with you what it looked like yesterday. So,
yep.
I can't move my own screen now. I'm like, what am I doing? Because like I'm like, oh, okay, that's me. So this was Roma tomatoes. It had just been picked. So normally, you know, tomatoes are picked green.
Mm-hmm.
So this was obviously towards this big for Roma tomato, right? So they had gotten a little bit late on these guys. This is kind of what the soil looked like. It was a little bit rocky. You know, soil type is probably going to determine this as much as anything. So this was, I mean, the pickers were actually all out here on this end. They'd already picked this section. And I was just kind of following them along.
Mm-hmm.
So that's kind of what we were looking at yesterday. So I took 4 samples.
Mm-hmm.
You don't need to take all 4 samples necessarily, but I took 4 just in case. So I took a soil sample about a trowel length away from the root system, right? Which mostly gets you outside the roots. And then I went 4 inches down on the trowel. Okay. And then I just pulled up and I took from the bottom that was stuck to here. Okay. So it was probably about 4 inches deep.
Oh, so just the base. You didn't get the whole core, basically.
I went for below surface. Yeah, okay. I didn't take an even section from across. I wanted to get stuff that was below surface.
Got it. Okay.
Because normally, I think we normally would want it, because I think we're trying to compare rhizosphere to something below surface. So it's
Yep. Okay. So that's a shovel length away, 4 inches down.
Yeah, trowel length away 4 inches. That's about a foot.
Okay.
You know.
So wait a minute. You dug the hole where the green hook thing is at the end of the handle.
That's how deep I went.
Okay.
Right, and then I didn't have a good. That's the last picture I have, I guess. It was hot. I was working by myself,
Okay.
and I had to get back to teach. So it was quick. But so basically, I went a trowel length down. I just then turned it up, and then I picked from the bottom of the trowel.
And is that your adjacent soil, or is that your rhizosphere sample?
That's my adjacent soil.
Okay.
So for the rhizosphere sample, what I did was I just took I took a shovel and I came in right in this area, sort of just adjacent to the root, and I just pushed the plant up, and then I took 2 samples because we wanted them for metabolomics and we wanted them for metagenomics, right? So I figured for the metabolomics, if we're looking at polar stuff, we probably don't want to put it into liquid, right? So I just took when you pull it up, right, a lot of the soil is going to come off the rhizosomes. Some of it's going to stay. So I just grabbed a section with the soil still on it, and I just cut it off with a knife, and I put that in a 50 mil tube, right? So I don't know. There's probably about like this much, maybe like 5 to 10 mils of soil total in that, and then a bunch of rhizomes. But I figure we can process it later. But that they can use that then as the soil sample for the metabolomics. Then for the metagenomics, I took another sample of those rhizosomes. I cut them off. I put it into some PBS, and then I just dunked the rhizosomes in the PBS to get all the dirt off of them.
Wait, what are you mean the roots?
Yeah, the
Okay.
thin roots. Right?
Okay.
And then so I dunked it in there to get all the dirt off of them. And like I didn't want big chunks I wanted dirt that was associated with the rhizome. So stuff that was pretty, not like it'll if you pull a plant out, right, it'll all fall off, but some of it's kind of a little bit more tightly in that rhizome. So that's what I wanted. Then I put that in there, and then I probably ended up again with like another 4 or 5, maybe 10 mils of soil, depending upon the sample. And then I took the rhizosomes and I put them into a separate tube. So in the end, I had a soil sample. I took about 15 mils of soil. That's outside the plant. I took a dry rhizosome sample. I took a sample where I washed the dirt off of the rhizosomes and put it into PBS. And then I took a sample where I put the rhizosomes that were washed into something else. So what that allows us to do, if we want to do it, it allows us to look at maybe 3 different metagenomes if we wanted to. We don't have to do the rhizosome. We don't have to do the actual root. We could just do the soil. That's fine. I think that might be in our best interest, is just to do soil adjacent and soil and rhizosome, because they should be different. And then also do we have plenty of soil to send for the nutrient analysis, right? That we were talking about doing, that John was talking about doing.
Yeah, elemental, 50 milligrams, so not
the elemental analysis, excuse me. And then we've also have a dry rhizosome sample and a dry soil sample that we can use for the metabolomics as well. So I think it makes the most sense to do the sample away from the plant and the rhizosomes soil sample for metagenomics and metabolomics. Those are the 2 that I would suggest doing. Does that make sense?
Why did you do the wet sample? What was that for?
To get the dirt off of the roots for the metagenomics thing.
But Joe's going to have to isolate DNA from that, right?
Yeah, but all you have to do is just spin everything down, take off the liquid, and then he can isolate DNA from it.
Okay, so we're thinking that the Oh, it's just you're just taking the liquid, so you're not actually extracting DNA from the soil.
No, he'll take the solid. Like we'll just take the tube, we'll spin the tube and pellet everything,
Mm-hmm.
and then we'll take the liquid off, and then he'll just have a wet soil sample, which is totally normal.
Yeah, but don't you lose some of the microbes in the liquid?
The idea was that hopefully when we centrifuge the thing, it should pellet the sample.
Okay. And we're freezing that? Because if you freeze it, then the microbes are potentially lysing, spilling their guts into the liquid, and then you can't capture that.
I did freeze it.
Okay.
Which is good. But that might, yeah, they need to be stored frozen. But yeah, we might lose some microbes that way. But
you have enough tissue or dry soil anyway, where we could just use the dry soil for both purposes. I don't know.
I mean, like, so they always could break. I think the most important, at least from what listen, it's always better if we can isolate DNA immediately.
Mm-hmm.
I think the most important thing from what I've seen is that if we're looking for differences between communities, we at least want to be consistent. So if we're consistent in what we do, then hopefully that should be the best we can do. Because, freeze-thawing any bacteria is going to have some kind of effect.
Okay. Okay, I'll collect exactly the same way then.
So, since we're getting close on time, if we can move on to the C Superb stuff.
Wait, before moving on, was there a question about number of samples? Is this just one plant at 2 different sites or multiple plants?
You did 4 plants, right, Nate?
I did like 12, but we don't have to use them all, right?
Oh, you did 12, okay.
And that's the beauty of having Nate submit the JGI form, is that he can determine, so you do need to know how many samples we're proposing to analyze or having JGI analyze. So that's all we need to know by tomorrow.
Exactly. Well, I also need to know how many to harvest. So should I do 10, Nate, something like that?
Well, no. So I don't think so. If we're gonna, I actually was thinking about that. So are we doing 3 sites or 2 sites? How many are we doing?
Right now, 2. The
2?
only places we knew tomatoes were growing.
So
yeah,
we've got 2. We got 2 different sites that we could use to sample off tomatoes.
Yeah. Do you have Romas by any chance?
They're not.
That's a bummer.
I know. They're good, but they're not Romas.
Yeah.
So how many plants, Nate, should we do?
So I would say, so if we do, so 2 sites, 4 times 2 for metagenomes and 2 for metabolomics. I think what our numbers, sorry, I'm just trying to remember. So let's see. So basically we're going to do, if they do between 1 to 92 metagenomes, and we're trying to split metagenomes with metabolomic polar analysis, they do 1 to 50. That means we can do around 25 metabolomic analyses and about 48 metagenomic. So if we're doing just 2 sites and we're doing 2 per, that means basically we would each get 6 plants that we could run total. Right?
Plants with 2 samples from each plant.
Yeah, because that would make 24 metabolomic samples and 24 metagenomes.
Okay, got it. And the
we could actually do, if we wanted to, we could also include the roots and do 36 metagenomes if we wanted to. I don't know if it's worth it.
Yeah, no, let's just stick with soil.
Okay.
Yeah.
So marching orders for JGI, look over, everybody, if you have time, look over the draft statements, make suggestions by tonight. Get your CV in if you want to be a co-PI. And Nate, if you've got any questions about this today or tomorrow, feel free to contact me independently if you need to for clarification or anything. And C Superb. So we've already been talking about title and abstract. Consensus at the start or near the start of the hour sounded like everyone was pretty happy with the way it looked. But again, we've got until this weekend. So everything's due Saturday night, please, to me. And so we've got time to revise the title and abstract more if you like. As far as team member contribution pages for all of us then, I've had a change of heart. We were all drafting our personal statements or whatever you want to call them on one of the Google Docs, but it would be better if you would move that into the individual Word document because we need to have each of those separately filled out by individual faculty members. I can't do it for you. So there is a Word document on Google Drive in the C Superb folder that says, I think, individual team member contribution page .doc or something like that. So we shared all of our personal statements on Google Docs so that we could see what each other were proposing to do, but you will need to fill out your own Word document and have it in by Saturday so that I can send them by Sunday.
So should we send that to you, Joe?
You can just drop it back in the Google Drive. After you filled it out, just add your last name to the Word doc or something like that. We'll just collect them all there.
Okay.
Or you can email it to me. Either way, it doesn't matter. I'll drop them in the Google Drive if you email them to me. So.
So one thing I wanted to ask about that, for what I was proposing to do was to develop a qualitative survey, a questionnaire.
Yeah.
And it seems like I'll have to put together an IRB for that. And that potentially can, I guess for me specifically, slow down the funding in a sense because I have to get that approved before moving forward, which is fine. So I can take care of that. So my question to the group is like, who would be involved in that part, the farmer interview component, the qualitative part?
Oh, definitely me.
Okay.
So yeah, it'll all be exempt though, right?
Right, but we need to, I think per the document, we have to submit that approval.
Mm-hmm.
Because we're going to do this as part of the human subjects for that part, at least.
Mm-hmm.
Yeah, we're just interviewing people and there's no risk to them, I think, so it should be exempt. But
well, is the interviewing, are you planning on doing the interviewing before the end of January? Because I think these contribution pages are only covering what we're doing between now and the end of January.
Yeah.
Right. So the question is, do we want that pilot data for the proposal in March? Right. So I would think that we will at least develop the survey and get it IRB approved so that we can collect data if we have time to do that.
Yeah.
But I think at least getting the instrument put together and also the IRB approval as a milestone as part of the.
Sure.
Yeah.
Yeah. So maybe that's what we work on is just getting the IRB. Maybe that's the contribution.
Mm-hmm.
Right. Okay. Yeah. So I just wanted to make sure for those that are involved in that part, that maybe we talk offline to coordinate that part.
Absolutely.
Yeah, I'd be happy to help with that too.
Okay,
For the IRB, do we just need the survey for that, or do we need the exact sites of who will be interviewing?
Yeah, that's a good question. I tried calling our IRB person yesterday, but they're out of town until tomorrow. So I'll figure that stuff out. But I think,
yeah,
regardless of that outcome, the goal is to put together and get an IRB approved, whatever the requirements are. But I'll try to do it through our site, but I know we might have to coordinate multiple CSU campuses.
Yeah.
Multiple.
Yeah, depending where we're sampling.
Yeah.
Yeah.
Okay. So anyways, we can deal with that offsite. So I just want to make sure we're including that as part of this, or if we should include that as part of it.
Yeah. Let me know if you have any other questions about IRB stuff too, because I've done a lot of IRB work here, which will be different from San Jose, but I can give advice.
Okay. Thanks, Joe.
Did you happen to learn anything, final details about the Superb submission then? We were going to talk about coordinating when we're going to show up in San Jose and maybe where we're going to stay so that each of us would know how much money to put on our request for travel and lodging specifically, not just travel.
Yeah. So the question about negotiated hotel rates, so the answer is no, at least based on talking to my chair.
Okay.
But we do get state government rates. So I don't know if that's something that you guys have looked into before. And only certain hotel chains would have that opportunity. So it doesn't save us a ton of money, but it might save 10, 15% off rates. So anybody have experience with that at all?
No, not me.
So certain hotel chains like the Comfort Inn and some other ones, La Quinta, I think, is another one that they have their state government rates. So as a CSG employee, you're a state employee, so you actually can get state rates for hotels. But that doesn't specifically answer the question. So, but looking around the options, you can, anywhere between like 150 to 250 is most of the hotels, I think, in the area. Yes, that at the maximum like 250, but it's probably gonna be less than that if we find something more reasonable. That there's a lot of hotels like really close by the Marriott that we're going to be at, so maybe looking at those first.
But at least it's much more reasonable than the 500 what they are quoting.
Right, it's a 560 or whatever it was.
Yeah.
And I checked, and they do not have a state government at that site.
So John, you may want to check with your procurement office on campus.
Okay.
Ask them for a list of state contracted hotels, because we do have that. And also, if you want to rent a car, CSU goes through enterprise only. So they do have state contracted rates. You just need to contact procurement.
Got it. Okay.
Yeah. We would need access to a conference room as well at this hotel, right?
Unless somebody gets a suite or
go to the Starbucks.
It'd have to be one heck of a suite.
Yeah.
I wouldn't worry too much about that.
Meet in the lobby.
Yeah.
Maybe. Yeah, we'll have to. Yeah, that's true. We'll have to figure that out.
If it's close to San Jose State, if we end up, we can always use something on campus, but right. That's to be determined.
Okay.
There's a hotel on campus, John?
No. We have on-campus housing, which is another option.
Oh, perfect. Yeah.
So if people are willing to stay in the dorms, that would be another option.
Well, even if they don't, if we could use San Jose State meeting rooms, that would
we can use meeting rooms on site. Yes, we have available conference rooms that we can use.
Okay. When do we want to show up, and how much time do we want to have before C-Superb starts? I think that's the last question for the day, in my mind.
What days are C-Superb again?
So normally the conference starts on Friday, but there's usually the Thursday of optional workshops. So question A is, we don't know what the workshop schedule is yet, I don't think, but I usually show up. I usually want to be at C-Superb Symposium on Thursday.
Yeah, I wouldn't be surprised if I have some kind of responsibility on that Thursday.
Yeah, I have the information for the Thursday, and the formal stuff starts at noon. So it's probably not enough time to just sort of meet for a few hours before C-Superb activity begin. So I'm thinking that Wednesday would be better.
Which means everyone's going to show up Tuesday night?
Or Wednesday morning.
Yeah. So decide what you want to do on travel, and either ask for 2 nights of lodging, Tuesday and Wednesday night, or just Wednesday night, depending on when you show up.
What's the date for that?
The 15th.
The 15th is Wednesday.
Okay.
And that would be the day that we would meet, presumably at San Jose State or somewhere.
Right. Yeah, it is winter break for everybody, so the campus will be empty.
Perfect. But you're able to get us into buildings, John?
I gotta run, guys.
Yep.
I'm gonna be somewhere pretty soon, so let me know if there's anything else that you guys need covered afterwards. And for now, what time is there a set time that I'm gonna expect to have everything in so I can submit it by Thursday?
Everything's supposed to be in tonight, midnight.
For the JGI, okay.
JGI.
Okay.
Yep, and Saturday for everything C-Superb.
Okay, great, thanks.
Yep. Thanks, Nate.
Bye, guys.
Bye.
Bye.
Good. So hopefully that gives everybody enough information to make decisions about how much money to ask for from C-Superb.
Maybe if we were to shoot for like Wednesday morning at 10 a.m., that would also allow folks to, if they want to fly in the morning, be able to accommodate.
Okay.
Get it, that could be the start time.
Sounds reasonable. So precise location to be determined, but San Jose State campus somewhere. Wednesday the 15th of January.
Yeah, 10 a.m.
That's the other possibility.
And any other questions? Does anybody feel like we need another Zoom meeting before the weekend? Be superb.
I don't.
Okay, hearing none, we're gonna say that we don't need one.
I think we're good, Joe. I think you've pushed us along quite far, so we have a pretty good.
I think if we wanted to look at the proposal, I'm sorry. Go ahead.
No, it's fine.
No, you go.
Oh, I just one question. So we asked for one night or 2 nights at the hotel because CSUPORP was paying for us to go to the meeting, right? Especially FCG members, Matt, do you know? Like, yeah, they're going to do the travel. So I just need to ask for the hotel, right?
Yeah, I emailed Susan yesterday and she said that since the travel wouldn't cost any more or less, depending on if you came a day earlier, the travel would still be covered by CSUPORP. It would just be the extra hotel nights.
Okay.
Yes.
Excellent. Thank you.
Yeah. Okay.
All right. Thank you, everyone. I guess.
Yeah.
Should we just? I guess we can go through Slack if there's any other questions, maybe.
Yeah.
But I think we're all good.
Perfect.
Sounds good. See you.
All right. Thank you.
Bye.
Goodbye.
See you, everybody.
Bye.
You are.
Bye, everyone.